Greetings students. Today we will talk about seeds. As you can see, I have collected a few seeds for you. So the first one, this one, these are gram seeds. These are fenugreek. These are flax seeds. Here I have some green bean seeds, and this is rice grain. Now, before we start with seeds, let me tell you that seeds, as we have learned earlier in the previous chapter, that seeds are ripened ovule. After fertilization takes place, the ovule changes into seed, or the ovule becomes the seed. Now, you must have noticed that while introducing these seeds, I mentioned these as grains and not as seeds, because these can't be called seeds, as these are actually fruit. These are fruit in which the fruit wall and the seed coat are fused together. Now, by fusing, they form a protective layer to prevent the embryo within. So there are a few more facts about seed that you should know. That it is a mature ovule after fertilization. All these seeds, they contain inside them the embryo, which can be a potential future plant. Now this embryo that is within these seeds, that's inactive or dormant till they receive a favorable condition to grow. And when they grow, that is when they germinate, these seeds, they contain the food needed for the embryo during germination. And these embryos that are within these seeds, they can withstand unfavorable condition like temperature, drought, and there are some seeds that are known to remain dormant even up to 100 years or more. Now, seeds can be classified into various types based on certain features. Broadly, the seeds are of two types based on how many cotyledons do they have. So, one is monocotyledonous seed, another is dicotyledonous seed. Monocotyledonous seeds are the ones which contain only one cotyledon, like this, like maize, like grasses, like corn, etc. Dicotyledonous seeds are seeds that contain two cotyledons. For example, a gram seed, pea seed, mango, etc. Seeds may vary in size. They can be as small as poppy. Their size makes it difficult to see them, through naked eyes. Some are as large as mango seeds. And do you know which are the largest seeds? Coconut. Coconut, they are the largest ones. Now, their shape and size may vary, but basically the structure remains the same. Remains the same means they will have endosperm, they will have embryo, they will have a protective covering. Now, on the basis of endosperm, seeds can again be classified. They can be called either albuminous or exalbuminous. Now, albuminous seeds are also known as endospermic seeds. In this case, the cotyledons persist. They are thin, membranous, but they persist. For example, a poppy seed, custard apples, cereals. And the other type is exalbuminous seeds. Now, exalbuminous seeds are the ones where endosperm does not persist, but the cotyledon stores the food for the plant, for the embryo. Okay, so you can see these are those exalbuminous seeds. Here, if you soak this seed in water, keep it for around a day or a few days, you will see that the cotyledons have become fleshy. That cotyledon, that fleshy cotyledon, is actually the place where food is stored for the embryo, for the seed, for the embryo that will germinate. So now let us go deeper into the structure of the seed. We'll talk about bean seed as an example of dicot seeds. Okay, so see, this is a dicot seed, as well as this, okay, these are also dicot seeds. Now when I'm talking about dicot seeds, beans fall under this category. For example, kidney beans, French beans. I only have green beans at home, so I'm showing you these. You don't need flax seeds here, so let me just move them aside. Okay, here you go. And here is our bean seed. Now you can see that bean seeds, or the dicot seeds, they have a kidney shape with one convex and one concave end. So this see, this is considered to be the concave side and this is considered to be the convex side. Now in this seed, if you notice, see, there is a scar. Can you see this white section? So this white section, or this oval whitish scar, that's actually the place, it represents the spot where the ovule was attached to the ovary through a placenta. This ovule is now the seed. So this ovule was attached to the ovary through a placenta at this spot. This greenish layer that you see, it's actually the seed coat. Seed coat consists of two layers, testa and tegmen, and both of their functions is protective, to protect the seed against any mechanical injury or any bacterial fungal infection. Now we come to the seed again. Here you can see that it might be a bit difficult to locate the pore here, which is easier in this seed, as this is dicot, so I will be moving back and forth once to this seed and next to the green bean one. See, can you see a hole here? Can you see this? This is actually the micropyle. This micropyle, as you know, is the opening through which pollen tube had entered the ovule, and this micropyle is the opening through which water enters, and seeds, after absorbing water through this micropyle, they germinate. Okay, the embryo, it germinates through this hole, this micropyle. This micropyle serves another function, that is, it helps in diffusion of respiratory gases, carbon dioxide or oxygen, for the embryo. Okay, now below the thick seed coat there are two cotyledons present in case of bean seeds, or in case of dicot seeds. So let us come to this. So you can see that these are two seeds which have started, where you can see the sprouts, you can see the embryo growing. So this greenish part, this is the seed coat which has come off because it is about to germinate now. It has started germinating now. So you can remove this easily, which has been removed from this seed. Yeah, which has been removed from this seed already. Now the two cotyledons I want to show you. See, these are the two cotyledons. Okay, the two leaf-like structures. These are the two cotyledons. Okay, these cotyledons, they store the food for the embryo. Now the embryo consists of two parts, the radicle and the plumule. Bear with me when I try to show you the radicle and the plumule. The radicle becomes the root and the plumule later becomes the shoot. So yeah, now let us check this out. Okay, so if we carefully observe this embryo that is growing, this part, the part I am pointing out right now, this is the radicle, and the one, this one is the plumule. So this part will give rise to the root, and this will become the shoot later on. Okay, and these are the cotyledons. Okay, I hope it is clear to you. Okay, now the plumule consists of a short stem with a pair of tiny leaves while growing. So that is what grows into the stem or the shoot. Now let me focus on the maize grain. Now this is small, and in these cases in this rice grain, the husk has been removed, the seed coat has been removed, and as I have told you that they are actually fruit with fused fruit wall and seed coat. So it is not possible, or in the span of this mobile's camera, to capture the micropyle and all in this. So for that I have a picture which I'll be using to explain things. It is of this corn, a kernel of corn. Now see, this is what the whole corn kernel looks like. If you look at this figure from here to here, you can see that this is different in color from the rest of the part. So this oval shaped structure is the embryo. The remaining portion is the endosperm. And see, here we get the seed coat. It is, I told you, seed coat and fruit wall, they are fused, so I can't identify it separately like the one that we could do in case of gram seed or in case of bean seed. That's not possible here. So if I do a cross section of this, it looks like this. This is the embryo, and this is the endosperm with the seed coat. Now I want to show you it in a nicer way. So see, the embryo contains the plumule at the upper end and the radicle at the lower end. Okay, now look at the endosperm portion. Can you see these cells? These are actually representation of the cells. This is the flowery endosperm, this is the horny endosperm, and these are actually the cells with starch granule in them. Okay, now the outer layer of the endosperm here, which is magnified here and shown as the aleurone layer, this aleurone layer is rich in protein. Okay, so the embryo is provided not just with starch but protein as well. Now the embryo consists of a single cotyledon that you can see. This single cotyledon is called scutellum here. Okay, this single cotyledon is known as scutellum here. Now the radicle is towards the pointed end, and it is, now the radicle which is present at the pointed end, it is considered to be enclosed in a protective sheath known as coleorhiza. See, this is the radicle, and this black U-shaped structure, it is the protective sheath known as coleorhiza. Okay, and the plumule that is present here, that is again protected by a sheath, this arch structure. This protective sheath is known as coleoptile. Okay, so basically that's what the structure of a bean seed and a maize grain is, or I can say what a dicot and what a monocot seed looks like. Okay, so that's it for today. We only have germination to talk about, which we will do the next day. Hope the structure of the seed was clear to you. You can ask any questions that you have in the group or in the comment section. Thank you, everyone.